I'm presenting here was a part of my PhD research, which allowed to identify for the first time the border disease virus in Tajikistan. Just not only in Tajikistan, but also in the Central Asia. It has been already confirmed almost around the world that pestiviruses are the frequent contaminants. I'm really sorry. I'm very sorry. That it has been already confirmed that almost around the world that pestiviruses are frequent contaminants of leaf-modified vaccines, the fetal serum, which induces infection in the organism of animals. This talk presents the results of the identification of the Hobi-like pestivirus in the virus vaccine against peste ruminants virus, the PPRV, which was used for the vaccination of small ruminants in some farms of the Republic of Tajikistan. As it is a vaccine congress I'm participating in, I've decided to present exactly this part of my PhD study and thereby emphasize the very urgent problem of vaccine industry's contamination. Hobi-like virus or pestivirus H is one of the 11 main viral species of the genus pestivirus in the Flaviviridae family. This virus was first identified in 2004 in a batch of bovine fetal serum and has been already confirmed to cause a natural infection in cattle and small ruminants. For livestock, particularly for sheep and goats farming, the greatest threat caused by the peste ruminants, which was determined as a transboundary and highly contagious disease of small ruminants by FAO and OIE. In this regard, in 2013, these two international organizations have jointly launched a global program for eradication of this disease in many countries of Africa, Asia, and Middle East. In Tajikistan, goat and sheep farming are traditional livestock industry. The breeding of sheep and goats are more profitable due to the fact that they are pasture animals and unpretentious to the condition of feeding and keeping. Livestock farms annually use the local and interstate pastures, which have a certain effect on the epizootic situation. Various breeds of these animals are bred in different regions. The Tajik woolly goat breed is very common among goats of the country. The main part of small ruminants of the Republic is focused in state and private farms. In almost all livestock farms in Tajikistan, diseases of the respiratory organs and the reproductive system of various etiologies are widely diagnosed. The peste ruminants is not an exception. This disease in Tajikistan, of course, was first discovered in 2004 by the common FAO, OIE, and local veterinarians conducted investigations. In our study, flocks of small ruminants from several districts of Tajikistan with symptoms of gastrointestinal, reproductive, and respiratory failures were examined, aiming to detect the pestivirus infections. In the flocks, mass abortion and stillbirth were noticed. The bronchopneumonia, nose and eyes discharge, oedema of the mucous membranes of the nose and mouth cavity, stomatitis, and diarrhea were observed during a clinical manifestation by the local veterinarians. A decrease in weight, growth retardation, and inability to stand independently were noted in the lambs and kids. In the second trimester, the pregnant evals were aborted. The fetuses were poorly developed. At necropsy, an increase in lymph nodes, liver spleen, and hemorrhagic pneumonia was detected. As I have already mentioned, the main objective of our study was to detect pestiviruses. In Tajikistan, data on pestiviruses are very limited. The identification and typing of viruses during mass outbreaks of diseases and the study of their etiology, particularly in sheep and goats population, is a relevant task of scientific research. So the above-mentioned facts determined the main goal of our research as identification of pestiviruses, mainly border disease viruses. Samples of serum and parenchymal organs of slaughtered animals and aborted fetuses was sent to our laboratory. The material was stored at -70 degrees and then examined for the presence of bovine viral diarrhea virus, border disease virus, and petits ruminants virus. The prevalence of pestiviruses was evaluated by agar gel immunodiffusion test, AGID, which is available, specific, and reproducible method. The test was performed by generally accepted methods, and as antigen, a clarified lizard of infected cells with reference strain of Oregon C24V of BVDV1A, the vaccine strain of PPRV virus were used. The test was performed in petri dishes in 1% lye of DIFCO agar and phosphate buffer saline. So to study the sera in an antigen was dropped in the central well and serum samples in the peripheral wells. The petri dishes were then stored in the room temperature, and after 24 hours were examined their precipitating lines. Isolation of total RNA of viruses was carried out by the standard method using a commercial RNA extract kit manufactured by Sintol Moscow, Russia, with according to the protocol. The reverse transcription PCR to obtain cDNA was conducted by the instruction of commercial kit of Sintol company also, and also the primers were chosen for a partial gene encoding NS3 of bovine viral diarrhea virus, 5 UTR, and N-Pro gene of border disease virus, partial N and F genes of petits ruminants virus according to the OIE recommendations. Phylogenetic analysis was performed in MEGA7 by the neighbor joining method with bootstrap values based on 1,000 replicates. In this slide, you can see the typical precipitation lines formed by using the BVDV antigen and the seras. The BVDV antigen is in the center wells, and the examined sera are in the peripheral. The detection of positive samples allowed us to use the further principle of test system proposed by Coggins for the diagnosis of equine infectious anemia. As you can see in this slide, we dropped the antigen in the central well, positive sera, and examined sera to the peripheral cells. So these are the control one, and these are the examined one. And obtained the positive precipitation lines. The results of the retrospective study in AGID has revealed that 27% of samples are positive to BVDV antigen. So the next step of our research was to examine the sample for the detection of antibodies to the petits ruminants virus. We used commercial vaccine as an antigen for the AGID test. The reaction scheme was the same as BVDV. PPR is endemic in Tajikistan, and young animals are more susceptible. According to the veterinary reporting data of Tajikistan, the morbidity rate of sheep and goats from PPR exceeds 40%. Accordingly, a comprehensive program for the eradication of the disease has been developed and included the immunization of livestock with imported vaccines. However, the experience of using some commercial biological products has shown their low efficiency, which can be explained by a number of reasons, including the reactogenicity of the products. In our study, while using PPRV antigens, we have detected that only 12% of the examined animals had antibodies to PPRV. In this slide, you can see the typical precipitation line with PPRV antigen. In some cases, we had noticed the formation of double precipitation line while using PPRV antigen. A detailed analysis of the results determined that some samples were positive with both BVDV and PPRV antigens, which composed 5% of the examined of the study serum. In this step of the research, we suppose that probably there is a co-infection with BVDV and PPRV virus in animals. Or the formation of the double precipitation lines may indicate the presence of another pathogen in the antigen. That is why we decided to test the PPRV vaccine. The results of reverse transcriptase PCR confirmed the presence of PPRV viruses in the vaccine. The comparative analysis with available strains in gene bank showed its homology. The subsequent analysis by PCR and phylogenetic demonstrated that the vaccine against PPRV was contaminated with the pestivirus H, the so-called Hobi-like pestivirus, or bovine viral diarrhea virus type 3, as some of the researchers would like to call it. It showed a 96% homology with the strain CH cahocoin isolated from isolated cell culture. We have deposited the identified nucleotide sequences to the gene bank under the accession number KX967, so you see 607. Thus, the contamination of a commercial vaccine against PPRV with Hobi-like virus has been established. We cannot exactly say whether the use of a contaminated vaccine has caused an infection in small ruminants. Due to a certain similarity of clinical manifestation in epidemiology of PPRV and pestivirus infection, the differential diagnosis of these diseases becomes complicated. It is supposed that the dissemination of pestivirus infection in the Central Asian region can significantly reduce the efficacy of anti-epizootic measures against PPRV. However, it should not be forgotten that the lack of proper control of biological materials of animal origin, such as cell cultures, fetal serum used in production of vaccines, creates a potential threat to the spread of causative agents of new emergent diseases. Actually, this was all I wanted to present today, and I would like, first of all, to express my gratitude to my supervisor, who was directly participated in the interpretation of the results of this work and my PhD work in general. I'm talking about the Professor Yuri Konstantin Pavlovich, who was one of the greatest virologists and unfortunately not with us today. And I would like once again to thank the organizers for giving me this opportunity to present my research in this Congress, and thank you very much for everybody for your attention.